Good morning, children. Next we are going to discuss about urine formation. So next we are going to discuss about urine formation. So in urine formation, it involves three main processes. So what are the processes? The first process is glomerular filtration, the second process is reabsorption, and the third process is secretion. Okay. So as the name suggests, I will just brief it first and then we will explain in detail. Okay. So what do you mean by glomerular filtration? Glomerular filtration means when the fluid, means the blood is moving into the kidney through the renal artery, thereafter it is moving where? It is moving through the afferent arteriole into the nephron, isn't it? So thereafter, after moving into the nephron, the afferent arteriole is branching themselves and forming a tuft of capillaries, which we call it as what? We call them as the glomerulus, isn't it? So thereafter you can find it is present, means this tuft of capillaries is being kept in a cup-like structure, this one, which we call it as what? The Bowman's capsule. Now this Bowman's capsule, when you see this Bowman capsule and this is the glomerulus, okay. So imagine this blood vessels, that is the glomerulus and the Bowman's capsule. Between them there are three layers. How many layers are there? Three layers. So from there, through the blood, the filtrate will be reaching the Bowman's capsule. Okay. So what are the three layers which are present? Listen very carefully. So this is the glomerulus. So the glomerulus is having a covering layer, isn't it? So they are having what? One layer. This blood capillary is made up of one layer. So that is one layer. Thereafter, Bowman's capsule, when you are seeing this Bowman capsule is having the second layer. So second layer is done. In between this glomerulus as well as the Bowman's capsule, in order to attach these two together, there is a membrane present, which we call it as the basement membrane. So basement membrane. So the blood has to get filtered. It is getting filtered through three filter papers. So what are the three filter papers? One is the covering of the endothelium of the blood vessels. Okay, so this is the blood vessel. Imagine this is the blood vessel, means that covering, that membrane they have to cross. Then there is a membrane called as what? The basement membrane. Then there is a membrane which is present in the Bowman's capsule, which we call it as what? The epithelial layer of the Bowman's capsule. So they have to cross these three layers and reach. So from the blood, the filtered, all what is getting filtered has to cross through the blood capillary wall. Thereafter they have to cross through the basement membrane, and then they have to cross to the Bowman's capsule's membrane, and then only they can reach what? The Bowman's capsule. Okay. So this process we call it as ultrafiltration or glomerular filtration. What is the advantage of this is that in the blood, what and all is present, all the impurities, everything is getting filtered. Ultra, as the name suggests, ultra means what? Much amount is getting filtered. All the substances, excess water, nutrients, hormones, medicine, what you are taking, everything is getting filtered excepting the proteins. Okay, the rest all materials, vitamins, okay, everything is getting what? The filtered, as well as the urea definitely is getting filtered. So since all the most of the particles are filtered, we call it as what? Glomerular filtration, or we call them as the ultrafiltration. Okay. Next is your tubular reabsorption or reabsorption. So what do you mean by reabsorption? I told you. So when the blood is moving into the Bowman's capsule through afferent arteriole, it is dividing into many tuft of capillaries called as glomerulus. So from there, in the Bowman's capsule, actually what is happening? The blood is getting filtered, isn't it? So it is passing through the three membranes and the blood is reaching the Bowman's capsule. Now when it is reaching the Bowman's capsule, listen. So it contains all the good as well as the bad materials. Everything is present. Now, what is the role of this thing? Next process is that tubular reabsorption. So what is tubular reabsorption? Bad things are going out of my body, it's good. But I am giving this much money, buying everything so difficultly. I am having my food, affording all the sort of nutrients, and everything is going out of my body. Whether it is good, it's not that good, right? So what happens is that after the ultrafiltration, the filtrate, what is reaching over here, it is moving through the tubules. First it will move through the proximal convoluted tubule, then it will move to the Henless loop, thereafter it will move to the distal convoluted tubule, then it will come to the collecting duct. So when this filter is moving through the tubules, I told you the blood after moving out of the Bowman's capsule, it will divide into efferent arteriole, and this efferent arteriole will divide into a network of capillaries. We call it as what? The vasa recta. So what happens is that through the vasa recta, when it is surrounding these what is that, proximal convoluted tubule, Henless loop and distal convoluted tubules, they are the blood capillaries are, efferent arteriole is dividing and surrounding them. So what happens when they are surrounding them is that they are nearer to these tubules. So this tubules, when you are seeing, when the filtrate is moving through this tubules, from the filtrate, the blood capillaries which is present, what the blood capillaries do is that they used to reabsorb, you know, they are taking out the good things, the excess water, the water which is needed, okay. So that and all salts, NaCl, needed good things, what and all is to be retained by our body. So that what they do is that they used to reabsorb. So that process we call it as what? Tubular reabsorption. So first filtration, everything has gone into the tubule now, mostly good as well as bad. From the filtrate which has been collected by the Bowman's capsule, which is moving through the tubules, in the tubules they are surrounded by what? The blood capillaries. And what is this blood capillaries doing is that they are reabsorbing the things which is needed by our body into the blood itself, okay. So that process we call it as what? Tubular reabsorption. The last process is tubular secretion. So moving on to this tubular secretion, in spite of reabsorption, this tubules, when you see this proximal convoluted tubule, Henless loop, distal convoluted tubule, all these tubes, what they do is that they do secrete something and put it into the filtrate, okay. So what they are secreting, you can find here K plus ion is secreted, H plus ion is secreted, okay. So H plus ion is secreted, NH3 ions are secreted. So like that, some substances are secreted by these tubules. So tubules are secreting something and putting it into the filtrate. So that process we call it as what? Tubular secretion. Tubules are secreting some of the ions and it is putting it into the filtrate. Now, why they are secreting? There is a question, isn't it? So why they are secreting? Listen carefully. So this is the structure of the nephron. I told you this is the cortex. This one is the outer medulla, this portion, okay, and this one is the inner medulla. Now, when you are looking into the structure, actually why they are secreting substances? There should be some concentration difference. Then only particles move from a region of one high concentration to a region of low concentration, isn't it? So, I have to take out something from here, which is very important. Imagine this tubules, from these tubules I have to take out NaCl. Here is given, this given, NaCl I have to take out, water I have to take out, okay. So I have to take out something from these into my blood capillaries, or it has to move out of the tubules, it has to come out, means I have to create differences in the ionic concentration. Ionic concentration means what? I have to give something here so that positive charge is becoming more and here it is becoming less, so that particles from here, higher part, will be moving into this. So something I have to take it out, like bicarbonates is very important, so I want to reabsorb that. But hydrogen, potassium ions are not that important. So I will be doing what? I will be giving something and supplementing those ions, ionic concentration, plus, minus, everything, so that substances can move out. There is no problem, no issues, okay. So in order to maintain, we call them as osmoregularity. What is that? Osmolarity. Osmolarity means only if the ions, salts, everything is present inside the fluid, from a region of higher concentration, water will be moving to the region of low concentration, okay. Along with that, so this fluid which is present inside this medulla cortex, we call it as what? The medullary fluid. In the medulla, what is present, we call it as what? The medullary fluid. So in this fluid there should be some ions, okay. So these ions, salts, NaCl and all has to be present, urea, have to move from one place to another means something has to be secreted by the tubules and given, so that good things, if it is going out also, the other ions which are not that important to our body is supplementing those things, that by concentration is maintained both inside as well as the outside the fluid, means between inside the collecting duct as well as inside the tubules as well as outside, the concentration is getting maintained. Okay, this we will be discussing in detail when we are discussing about mechanism of urine formation in detail. Now we are just seeing what we have discussed once. Okay, so first is what? Glomerular filtration. So the filtration of the blood which is carried out by the glomerulus, we call it as what? Glomerular filtration. You can see in a minute, okay, per minute around 1100 to 1200 ml of blood is filtered by kidneys. Okay, by the kidneys per minute, this much is filtered. Okay, so I told you glomerular capillary blood pressure. So there is a pressure, you can find these since there are much amount of capillaries, the blood is moving through one vessel, and thereafter you can find many blood capillaries are present, so there will be very high pressure there. Okay, this pressure is helping in filtration parts. Okay, so they are helping in what? Filtration. So causes filtration of the blood through three layers. I told you, one is the endothelium of the blood vessels. So the glomerular blood vessels, the endothelium is the first part. Second is the epithelial layer of the Bowman's capsule and the basement membrane. So the blood capillaries, first membrane endothelium, Bowman's capsules membrane, in between there is a basement membrane. Through these three layers only what has to pass? The blood has, the filter has to part. From the blood all the substances are getting filtered. Okay, the epithelial cells of the Bowman's capsule. Now to your information, the epithelial cells of the Bowman's capsule. So if you are seeing the Bowman's capsule, the epithelial cells of the Bowman capsule is having small pores. These pores we call it as podocytes. What do we call them? We call it as the podocytes, or the filtration slits through which the substances are getting filtered. Okay, so that you have to remember. Podocytes arranged in intrinsic manner, integrate manner means inside. So they are leaving what? Minute spaces. Okay, pores like structure called as filtration slits or slit pores. Blood is filtered so finely through these membranes. Okay, that almost all the constituents of plasma, you know, fluid part of the blood, all the constituents I told you excepting the protein is getting filtered, okay, into the Bowman's capsule. This process is called as what? Ultrafiltration. So per minute how much amount of filter is produced in the Bowman's capsule is called as glomerular filtration rate. So what is GFR? It is glomerular filtration rate, which means per minute how much amount of filter is collected in the Bowman's capsule. Okay, the second is what? Tubular reabsorption. So you know, nearly all the essential nutrients in the ultrafiltration is getting filtered, isn't it? So it is reaching what? The Bowman's capsule. 70 to 80% of electrolytes, water, everything is reaching what? The Bowman's capsule. So when through the Bowman's capsule the filtrate is moving through the PCT, okay, maintaining the pH, ionic balance of the body fluids. Okay, so we have to maintain that one. So they are selectively secreting what? Hydrogen ions, ammonia, potassium and all are secreted and put into, and they are absorbing what? Bicarbonate ions from it. So in order to maintain the balance between the inside fluid as well as the outside blood, medullary interstitial fluid blood, they either secrete or they reabsorb. Okay, so like that reabsorption is also taking place. Henless loop, if you see, this region plays a significant role in maintaining high osmolarity of the medullary interstitial fluid. So the descending lymph of the Henless loop is permeable to water but almost impermeable to electrolytes. So the descending lymph is permeable to water but not to the salts, but not to the ions. So you can find here when the filter is moving through this membrane of the Henless loop, descending lymph. So this is descending lymph, this is ascending lymph. Okay, so the descending lymph of the Henless loop, when you are seeing, they are permeable to water but not to electrolytes. Electrolytes means what? The different ions. Okay, to that they are not permeable. Salts, they are not permeable. They are permeable to only water. So water from here will be moving out. Understood? So water is moving out, and what is there inside? The salts, the electrolytes and all will be present inside the loop. Okay. So here when it is passing down, actually the urine become more concentrated. Why it is becoming more concentrated? Because salt is not coming out, water is coming out. So water is less. So dissolving capacity is lost. So the salts and electrolytes are becoming more. In that case, what happens? It will become hypertonic, isn't it? So here when it is coming down, the urine will become more concentrated. But when it is moving up through the ascending limb of the Henless loop, the permeability changes. The permeability means what? Allowing what to come in and what to go out changes. Now these walls of Henless loop, what they do is that they will allow salts to move out, but not water. So water will be retained, but what can move out? Salt can move out. Now when the salt is moving out, the salt concentration is decreasing. So what will happen? The urine will become more dilute. So when it is moving out, you can find the urine is becoming dilute. Here the space is increasing, which means that they have become dilute. So when the urine, filtrate is coming down through the descending limb, since water is moving out, it is becoming concentrated urine. When it is moving up, water cannot move, only salts are moving out, so they become dilute. Okay. So that is what we have to look into Henless loop. So in this region, so in case of proximal convoluted tubule, what happened? Proximal convoluted tubule, both secretion is also taking place and absorption, reabsorption is also taking place. In case of Henless loop, what is taking place? Here, the descending limb of Henless loop is permeable to water, which means they will allow the water to pass through the membrane, but they do not allow electrolytes. So it concentrates the filtrate. It is becoming much concentrated. But the ascending limb is impermeable to water, but it allows transport of what? The salts. Actively or passively they can move. Therefore, the concentrated filtrate pass upward, it gets diluted. Okay. The electrolytes are moving out, water is there, that's why it is getting what? Diluted. That is what you have to note when coming to Henless loop. Now, distal convoluted tubule, what it is doing? Here also reabsorption is taking place. Sodium ions which are present inside the tubule is getting reabsorbed, water is getting reabsorbed. Okay, reabsorption of bicarbonates HCO3 minus is taking place, and they are secreting. This distal convoluted tubule is doing secretion. What they are secreting? So proximal and distal convoluted tubule both do reabsorption as well as secretion. Some substances is secreted, some substances reabsorbed. Okay. In order to get some substance, I have to give something and get the other one, right? So here selective secretion of hydrogen. So hydrogen is secreted, potassium ion is secreted, NH3 is secreted, ammonia is secreted. Okay, so why they are doing? In order to maintain the pH, acidic or basic pH has to be maintained. So this is done with the help of sodium potassium pump is a mechanism which you will be learning in your higher classes, right? Next, moving on to collecting duct. So today what we have discussed about ultrafiltration done, tubular reabsorption and secretion is taking place here. Here the concentrated urine, dilute urine. Here also you can find what? The tubular reabsorption and secretion is taking place. Then the urine is now reaching, the filtrate is now reaching the collecting duct. In the collecting duct, what is taking place? That's what we are going to discuss next. So in the collecting duct, large amount of water could be reabsorbed. Okay. So water is not let, especially when it comes to summer and all, we lose water through sweating, isn't it? So in that case, water is needed more, and I am not drinking much water means automatically you can find the urine will become much yellow. Why? Because it is becoming highly concentrated. Why? Because here the medullary fluid is present now. So the fluid which is present over here in the kidney, the medulla. So that fluid, what they do is that they help in reabsorbing the water. Water is getting reabsorbed. Okay. So that what happens is that the thing is what the urine is becoming much concentrated. This segment allows passage of small amount of urea into the medullary rate. I told you urea is there in the collecting duct. Some amount of urea will come to the medullary fluid. Why it is coming? I told you again, to maintain osmolarity. Okay, to maintain the pH. It also plays a role in the maintenance of pH and ionic balance. So that is the main importance. pH means what? Hydrogen ion and hydroxyl ion concentration, positive, negative ion concentration. Okay. So ionic balance has to be maintained. So they also, this collecting duct also secrete H plus ion and K plus ions. H plus is hydrogen and K plus is potassium ions. So there are three processes: tubular reabsorption, tubular secretions. So tubular secretion, tubular cells secrete substances like this ammonia into the filtrate. So this is why they are doing it, in order to maintain the ionic and pH balance, that is acid base balance in the urine. Okay. So with this we are coming to the end of the part what we are discussing today, the chapter, that is urine formation. I repeat, so in the urine formation there are three processes. One is glomerular filtration, reabsorption, secretion. All things are filtered. From the filtered, good things are reabsorbed. Okay, needed things are reabsorbed. In order to reabsorb that, something has to be secreted and given, so that only they can take the things which is needed. So that part we call it as what? The secretion. So why this reabsorption and secretion? Some substances are reabsorbed, some substances are secreted. Why it is needed for secretion and all? I told you, in order to maintain osmolarity, concentration of water, in order to maintain ionic balance, in order to maintain pH balance. Some substances are secreted and some substances are reabsorbed. Okay. So to reabsorb something, I have to give something. So that is what taking place during secretion. Understood? So that's why the tubule has to secrete something. With this, we are coming to the end of the portion, urine formation. In the next class, we will be discussing about the mechanism of your urine formation. Okay. So how the concentration of the filtrate is done, mechanism, that in the next class we will discuss. Hope it's clear. Thank you.